So I'm going to tell you this morning about some of the processes we've been running to identify the most critical resources in life sciences. And it's been something that's been in the pipeline for nearly a decade, so I hear. I've only been involved in the last few years, but it's really been a lot of work by a lot of people, and it's really culminating now in this process we now have to identify the critical resources in life sciences from a European perspective at least. But as we know, a lot of these resources are also international, so we'll start with Europe though. So open data, we all rely on open data to do our work in bioinformatics, and more broadly, biologists rely on open data every single day to do their work. And this has been recognized by a number of initiatives and policies from bodies such as the European Commission and the G8 group. Additionally, the Royal Society a couple of years ago published a Science as an Open Enterprise paper which extensively supported open data for science, for doing science and moving science forward. But of course this requires infrastructure. It's very difficult to manage and reuse open data if it's all over the place on people's laptops and goodness knows where. So in the life sciences we're quite fortunate, if you like, that we have a number of data resources already existing that help us do our work, and we can build on that to make a more robust infrastructure for the future. I just thought I'd take you back 20 years. At that time I was working at a journal called Trends in Biochemical Sciences, and we had a column called Computer Corner, which we thought was very trendy because it was about how you use computers in biology. And this struck me as interesting, as in those days, scientists would focus on the things that they were interested in and make a little website. As you can say, it even says Uniform Resource Locator, not URL. So it's relatively early days of the internet, and all these resources now, at that time, you can see on Stanford, Harvard, Arizona, all over the place, but all at universities. And this is the kind of first-generation website one might expect to have seen. It looks quite dated these days, and of course many of these websites went into decline, and this one here, for example, it's flatlined in 2002. So that's where we've come from the last 20 years. Where are we going in the next 20 years? So when we want to talk about data infrastructure, we need to know what data we're talking about and what resources we're talking about. So how are we going to identify those critical pieces? We need indicators. We need metrics and other more qualitative measures of what is important in order to identify them and then understand what we're talking about. And we need that so that we can talk to funders and other influential bodies about how we want to run these resources in future for the betterment of science. The users need to understand, technically if they're looking at a useful and robust resource. In terms of ELIXIR, there's different countries have different capacities at the moment and expertise, and we see that ELIXIR is a very important way through the core data resources of transferring knowledge and improving standards and excellence all round. Finally, speaking as someone who runs one of these resources, you need to know these things in order to understand how to take that resource forward, and taking that resource forward could be expanding it, or it could even be retiring it. And you need to understand the scientific trends so that you can see what resources need to be built, as well as which ones perhaps are no longer required. Okay, so what are the ELIXIR core data resources? We have defined this as a set of data resources that are of fundamental importance to life science research. And together they provide a collection of resources that are of generic value to life sciences and have high levels of usage and a high quality and high technical performance and so on. So after a lot of discussions, we came up with a set of indicators, and we basically put them into 5 different categories. The categories are scientific focus and quality, community served, quality of service, legal and funding infrastructure, and impact and translational stories. And together, when you put them all together, you can then make decisions about where that resource lies in the ecosystem. So this is not like a score that we're making here. We're giving people information to make good judgments on things. We'll go into the indicators in a bit more detail. So first of all, we need to know, is this database an archive or a knowledge base? So is it something that people submit data to, the community submit data to, or is it a knowledge base which is a curated resource, typically, for example, looking at scientific papers and extracting information, or potentially computing on top of the submitted data in order to make a nice or more easy-to-use interface. We need to know what the scope of the resource is, how comprehensive it is. Is it across all species? Is it just one species? Is it a protein family or all protein families? How international is the database? Is it generated from an international effort? Who funds the database? Is it just one source of funding, or is it multiple sources of funding across the world? And what is the staff effort? Here I've put curational effort, but of course bioinformaticians and developers are also part of the staff of many of these databases. And this just gives you an idea of how, what the effort is to generate this resource. And in terms of the community served, obviously one of the major quantitative metrics is access to usually a website, but also other methods like APIs and FTP sites. And it's useful to know what the potential usage is, and this is really a sort of protection, if you like, for databases that have, I wouldn't say have a sort of specific user group that might not be enormous, but it will show then how critical that resource is to that user group. So, for example, if there are only 600 labs using a particular kind of data in the world, and you have all 600 labs using that resource, then obviously you've got a tremendous coverage there. But 600 IP addresses is probably not enormous by comparison to other more major resources. That doesn't necessarily mean that smaller resource is less important. It just means it's different. And this is why the human judgment is important. Another quantitative metric is the usage in the literature. So there's three types of things you can measure in the literature. One is citation of accession numbers. The other is mentioning the resource. And the third one is citing the paper, the NAR paper typically from the database issue. How many papers cite that, those papers about the resources. And finally, another really important point is the dependency on the database by other resources. So this is a very, very typical thing in bioinformatics, as we know, is that this data is very fluid. It moves in and out of different resources to do different things. An example of this is Uniprot, where clearly Uniprot is reused a number of different other more specific resources. And if you just measure the hits to that one resource and ignore all the other places where that data is reused and found, then you're going to underestimate the impact of the resource. Quality of service. Obviously, we need to, it's not my laptop, so I'll just get rid of that. Persistent identifiers. Obviously, the rule 101 of bioinformatics is that each object in your system needs to have a persistent and unique identifier. If it's a deposition database in particular, what is the throughput? How many entries? How many depositions are made a month? What's the uptime and performance? Obviously, something has to be reliable. Does it use community standards for metadata? Is it clear if there are facts or assertions in the database? Is it clear where they came from? Does it link to papers? Are other means of accessing the data available other than just a website? Are there APIs? And if so, what kind of outputs do those APIs generate? And does it have a customer help system, such as a help desk or mechanisms of feedback and user research? This is a really critically important set of indicators, which really is about how the governance and legal and funding infrastructure. Typically, these resources, core resources, will have an international scientific advisory board, and there should be, really important, a legal framework that supports open science, which basically is talking about licensing and the ability to reuse the data. And you should have the usual things like a privacy policy and an ethics policy that are clear, and be able to demonstrate funding has been achieved for a period of years and is also available going forward. finally, this is probably one of the hardest categories to talk to, which is the impact and translational stories. And this is really to try and show the wider impact of the resource beyond the research community in ways that a non-bioinformatician might understand. So one example here might be the reuse of those resources in patents, really branching out into a more industrial and commercial setting. Right, so that's what we did. I'll come into the process in a minute. But just to mention FAIR, we did benchmark FAIR against the indicators, and they map really quite well. Most of the FAIR indicators are mapped to the sort of more technical side of things, so in terms of the data availability and metadata standards and so on. And I think probably the difference between the indicator set that we've come up with versus the sort of FAIR indicators are that the core data resource ones address impact and community need, in a more comprehensive way than the FAIR ones do. And they are supposed to support decisions on value, not just be a score. So if you want to know more in more detail about all these things, or digest it in a different way, there's a Factor 1000 research paper on the ELIXIR channel, which was updated relatively recently with all these indicators in it. And the process, which I'll come to, I'm not going to talk about this too much, but we have run a process in which we asked people who ran, via the heads of nodes of ELIXIR, people who ran resources that they thought would be core. So this means that in ELIXIR speak, there's a node, and the node has a service delivery plan. In the service delivery plan, there'll be data resources, and then the node would run a process of some kind to put forward some of those services as core data resources. And then we had a little form where it's relatively straightforward to fill in a form where you'd fill in all these indicators and the values of those indicators. And then these were reviewed by both heads of nodes and external reviewers. So it's a fairly robust and broad approach to doing this. And I think 8 reviewers each reviewed the applications, and a score was assigned of 1 to 5 for each indicator group, and then this was normalized. And then this was discussed at a heads of nodes meeting back in February. And when we started looking at the nitty-gritty of these things, as typically happens, it all sounds quite good on the surface, and then you start actually doing it, and you find there are other things that you hadn't really thought about, or things that were difficult. And these were roughly in 3 different categories. One is scope. We struggled when we were looking at these things to define, what kind of scope is important. And in the end, I think the thinking is that, if you're a deposition database, then your scope is generally, is broad, and it refers to the type of data. But if you're a knowledge base, the scope can be anything from comprehensive to something quite narrow. And I think the comprehensiveness becomes quite important if you're a knowledge base. I'll just skip to the third one in a moment, because the middle one actually is probably the most difficult in some ways to continue to work on. But the licensing became a question. And what we'd really like to see is something like a terms of use that says everything's in the public domain, or a Creative Commons license that support open science, and those would be CC0, CC BY, CC BY SA. But we saw quite a lot of different licenses, and we've had a really strong position on open access and open science. So these things needed to be resolved for some resources. And finally, the nested resources proved particularly complicated, and I don't think we are quite at the end of this discussion yet. In fact, probably only at the beginning of it, where lots of resources come together in a collaborative way to contribute to a larger umbrella resource, for want of a better word. And a good example of this is the Interpro Consortium, where lots of different databases all over the world are pushing content into Interpro. And trying to figure out the relative credit that should be given to all those different partners is tricky. So I'm happy to announce now that the initial this is the initial set of core data resources that we identified through this process. And I say initial because we've just done this once. We've learned a lot from the process, and we'll be running it again. And you'll note the really important thing to note here is, I'm sorry for all the acronyms, I'm assuming this audience know all these acronyms, is that it's not just deposition databases, this is also knowledge bases that are in this set. And going back to the Interpro example here, we've just listed Interpro, but it's really, really important that all the contributions of all the constituent databases is recognised, and we'll be working on that further in the future. Another thing that came out of this process was the need to really define some deposition databases. As I mentioned, the core data resources are a mixture of resources, but increasingly the ELIXIR Hub get questions from funders and journals. I work with a lot of journals, and they're always asking me this as well, is what do we recommend as the deposition databases? When people write papers, they have data, obviously, on which those papers are based. Where should that data go? And as probably everyone's aware, many funders are now really revisiting their data policies and formulating data management plans. So this question comes up over and over again. So through this process, we thought we needed to have also a list of deposition databases with which we can talk to funders and journals and so on, anyone else who wants to know. So how do we define a deposition database? Well, firstly, it needs to be part of an ELIXIR node service delivery plan. That's critical. And then it must accept experimental data, from a broad international community of researchers and others, potentially, that are beyond the funding envelope of the database itself. So you can't just make a database and then submit to it yourself and then call it. It has to be serving a community beyond your own group. And it must really meet some of the technical, certainly the technical and governance criteria that you would expect of a core data resource. But it doesn't have to be up there as a core data resource. It might be at an earlier stage of development. As we know, science moves, a new technique comes out, a new type of data is generated. The community realise they want to put this all together in one place, manage it in a collective way with standards and whatnot, and that might be at an early stage of doing the science. And in the fullness of time, we will see if that kind of resource becomes of critical importance. But at the time, you want to encourage depositions to that database. You want that to be on the map, but it might not be fully at the level of a core data resource. So it doesn't have to be a core data resource. So here's our list of deposition databases. Again, it's an initial list, and these cover the whole scope of life sciences as far as we know. Again, we're very interested to hear of other deposition databases. This is the best we've done at this point in time. And this list will grow and it'll potentially shrink to reflect scientific requirements. So for my last slide, we will have another round of this core data resource process next year, and we'll incorporate all the learning from this initial round that we did. We're going to start a forum of the people who run these resources in the data platform so that we can really start to put a tangible shape on what it means to have all these data resources considered as an infrastructure. So that means we need to discuss what kind of statistics or user statistics we will publish and how we are going to describe this thing as a whole going forward. And then finally, there's a sort of, I won't say parallel activity, which is this Global Data Resource Coalition, which recently published a paper, a paragraph in Nature, pointing to a more full position paper in bioRxiv on how we're going to coordinate internationally to support core data resources. And I think the ELIXIR experience is really helping to inform that, and that will be taken forward over the next few months. So that's all I have to say, and take any questions. Thanks.
Thank you very much, Joe. Any questions for Joe?
Hi. Biocore DB was a sort of community-driven effort to kind of define and do a lot of these things. So does this build upon that or replace that, or have you been working with the people that originally set that up?
I actually have not heard of biocore DB, tragically. So what Who did it?
It was from the Biocuration Society like years ago.
Okay.
So,
Well, I wouldn't say it built on it, but we certainly should have a look and see what they've got there. I mean, certainly we've involved lots of curated databases.
I'm sure lots of those people were involved.
Yeah,
so.
no, thanks. We'll have a look.
Hi, just a quick comment on your community usage. In some recent work done on the usage of major model organism databases in particular, they're not cited in the literature typically, but they're heavily used. And we can see that disparity when we look, we are looking at measures of capturing their usage sometimes in the acknowledgements, but often not. And it's an immediate comparison there is with gene ontology, another major resource where it's often mentioned in an article because of the use of that particular resource for data analysis, in particular for gene enrichment analysis. So I wonder if you take, in the future, I would love to think working with publishers where the publishers start capturing the metadata about which resources were used, we'll get a better method of capturing usage in that way.
Yeah. I couldn't agree more. I mean, I think it's it's one of the reasons why we have a number of indicators, because if you just, focus on one, like citations, there are a lot of really, really important resources that would get underestimated by just that measure. And I think, having a sort of cohort of resources, I know that some of the mods are, we're working on a more international view of these things, but I mean, once we can talk about these things as a whole, I think it'll make a much more coherent conversation with publishers. I feel that there are many very good journal editors out there who really care about these things, but it's very difficult for them to, have sort of 47 parallel conversations, each one about a different resource. I think the more we can come together to make a coherent argument, the more likely we are to see resources cited better in the literature.
There are no more questions for Joe. I'd like to thank you very much.